Quentin, if you're ready to begin, you can go ahead and advance the slide to your poster.
Alrighty. Hi, everyone. My name is Quentin Gautier. I'm a PhD candidate working for Dr. Bruce McCord here at Florida International University. I've been here for about 4 years now, actually nearly 5 years, and uh, the focus of my research here at FIU has been for using DNA methylation patterns for forensic applications. Primarily, what I've been doing is looking at body fluid identification using DNA methylation patterns of various markers, as well as looking at the ability to predict age in an individual based off of other DNA methylation patterns. So, to be brutally honest, I'm not 100% sure how to go about presenting a poster here without really seeing or hearing anyone. So, I'm just going to give a brief overview of the poster. And if anyone has questions, I'd be more than happy to try and elucidate anything I might have missed. So, the basic premise of this research is that, you know, uh, creating STR profiles, we can get random match probabilities, likelihood ratios that are, you know, such a high degree of certainty that it's pretty much guaranteed that a good single source profile can be sourced to an individual. However, what happens when there is no suspect and there are no hits in our databases? Well, some people have turned to various phenotypic assays that are going to be able to predict things such as hair color, eye color, skin color. And obviously, we do have the ability to determine someone's gender based off phenylogenin. However, with that assay, we still don't know anything about the individual's age. So we have been trying to focus on predicting age as one of the more useful aspects when there is simply no suspect, no leads for investigators. In our lab, we've been working in the past few years on body fluid identification and age determination using DNA methylation. The reason that we are so focused on those two aspects in conjunction with each other is that research time and again in our laboratory, in other laboratories that we've collaborated with in Europe and in Asia, have shown that the uh, body fluid that a DNA sample comes from can influence the accuracy of these age models. And in fact, there are different age models or different markers in the human genome for DNA methylation that work better in one body fluid versus another. Previously, I've been working on creating a body fluid identification multiplex, and that was done using Pyro Sequencing in our laboratory. Now, Pyro Sequencing offers a quick and cheap method for these analyses, but doesn't allow for realistically more than 4 regions of DNA to be amplified together and then sequenced without running to some serious issues. It is a technique that is really, really straightforward and easy to implement. However, it does have a few shortcomings in the long run. So, what we wanted to do is uh, start looking at moving this towards a larger, massively parallel sequencing platform, such as the MiSeq FGX from Illumina. The idea in this project is that we're going to be able to amplify nearly 20 regions or 20 markers of DNA that are going to be looking at body fluid identification for saliva, blood, vaginal epithelia, and semen, as well as age prediction models with saliva, blood, and semen. There don't there are not very many or any at all vaginal epithelial age prediction models out in the literature as of yet. It has been a marker that is very elusive to find a good predictive model. Once we have identified the regions of DNA that we're going to be looking at, and this is based primarily off of the body fluid and age markers that were developed in our lab, as well as the age markers that were developed in our collaborators' labs around the world. We're going to be able to amplify them in a single multiplex reaction with absolutely no uh, markers on our primers. So we don't need any fluorescent labels. We don't need any biotin or anything like that. So it allows the PCR aspect of this to be extremely cheap. You know, these PCR primers are only going to cost us about $10 for thousands and thousands of reactions. From there, we're going to be evaluating just how well our PCR amplification is working, and we're going to be able to use a kit from Illumina that quickly prepares PCR amplicons for sequencing on their sequencer. So to get a little more specific on the method, looking over here on the right, so the samples obviously can be collected from crime scenes and extracted using, for example, in our lab, we use primarily the QIAGEN EZ1 DNA Investigator Kit and quantified with qPCR on the rotor gene Q or with a qubit. Once we know the quantity of DNA in a sample, we can convert it using the QIAGEN EpiTech Fast Bisulfite Conversion Kit. The purpose of this kit, and I'm going to be able to show this a little bit better actually on this slide here. The purpose of this kit is that we're able to modify unmethylated cytosines into uracil using this bisulfite conversion, while the methylated cytosines are going to remain unchanged. What this allows us to do when we're doing our PCR amplification is uracil is going to be base paired to adenine and then back to thymine, whereas our methylated cytosine will remain base pairing with guanine back to cytosine as normal. As I said, we want to evaluate on the amplification efficiency of this multiplex. And so initially, trying to get this amplification going, it's going to be on the Agilent 2100 bioanalyzer. And what this is going to do, this is a CE on a chip instrument that's going to allow us to visualize the peaks for each one of these amplicons that we're coming up with. And also give a relative quantification of the sum total of DNA in that sample. So this is going to let us know, is our amplification working? And how much DNA do we have after PCR? That knowing how much we have is going to be important for going on to the next step, which is library preparation. And that is with Illumina's Nextera XT DNA Library Preparation Kit. This is a very fast library prep kit from Illumina. The protocol takes only about 90 minutes, and that's with about 15 minutes of hands-on time. The general concept for this is you are going to be taking DNA. It can be for genomic DNA and for PCR amplicons. And you're going to be using Illumina's transposome. So what this is doing is simultaneously fragmenting and tagging fragments of DNA that are going to be approximately 300 bases in length. And so in this center area is going to be the regions of DNA that we're most interested in for determining body fluid and age. The rest of the protocol is simply getting some sequencing primers base paired to our prepared fragments of DNA. And all this is doing is getting our region of interest prepared for sequencing on the MiSeq, where we then use the MiSeq FGX. And if anyone's ever gone to an Illumina presentation showing about how their massively parallel sequencing platform works, it's the same general concept. The bridge amplification so that we can get bidirectional reads of our region of interest, and then sequencing. Once we've finished the entire process of getting our data, that's where it's going to become a little bit complicated. So what I envision is that a lot of this DNA is going to be presented as each region that we're interested in. We're going to align all of the reads. And at the CPG sites, these DNA methylation sites that are helping us to determine either body fluid or age, we're going to see this ratio of thymine and cytosine. And so we're going to be able to determine the percent methylation at that location based off of the number of cytosines over the total number of reads for that sample at that location. From there, we're able to go on to data analysis. And so I want to hop over to here. So the data analysis for the body fluid identification. So this has been completely done in-house. The body fluid profiles that we've come up with for saliva, blood, vaginal epithelia, and semen are such that we're able to generate a profile based off of 4 markers, 2 CPGs in our blood marker, 1 CPG each from the saliva marker, vaginal epithelial marker, and semen marker. And this right here gives an idea of what a semen sample, the results that we would get. So about 7% methylation across the first 3, a little bit lower on this third one. Nearly 80% methylation on our vaginal epithelial marker, and then 8%-ish on the semen marker. It's very hypomethylated for semen on the semen marker. We're then able to use a process in RStudio. It's a mathematical prediction formula called tidy LPA. So it's latent profile analysis. This latent profile analysis allows us to create profiles of what is saliva in the context of these 5 CPG sites, and then the same for blood, vaginal epithelial cells, and semen. The end result is that once we've created a database and trained our model, we're able to input an unknown sample. And given these percent methylations that you see at the 5 CPG sites, it will give the posterior probability that that sample belongs to one of the 4 profiles corresponding to a body fluid. And so you see we can get very, very close to 1 probability, basically 100% for saliva, and the probabilities that it could be the other body fluids remain extraordinarily low. So once we know what our body fluid is, we can move on to the age prediction. And so this is where there's going to be some variation for, depending on the body fluid, which methylation marker sites we're going to be looking at. I do have an example here from our collaborators in South Korea, Dr. Lee, the age prediction for semen samples. And so this relies on just 3 markers. And once you've predicted the percent methylation in these 3 markers, plug it into a formula, and it's going to give you a predicted age. And so in the paper that they published a few short years ago, they were able to create a training set with a fairly good correlation with a linear regression model, and then tested that set. And as you can see, the predicted age is pretty darn close to the chronological age in most cases. And so that's going to give investigators some kind of lead, whether that be for the exclusion of certain suspects or the inclusion of suspects. In particular, with labs getting interested in genetic genealogy. Say, for example, you've run an unknown STR profile through a genealogical database, and you have reason to believe that it is a male from this family, but you have no idea if it's the father or the son. Well, if you had an idea about what age the person is, if they're supposed to be in their 50s or in their 20s, you would be able to focus a little more easily on one of the 2 individuals. So this project was initially conceived in the fall and was a recipient of the IFRI Student Support Grant to work on it starting this spring semester at FIU. I got to the point of designing some primers and identifying each of the regions that I'm going to be using for the marker sets, but I did not have time to actually start a lot of the process before the coronavirus pandemic hit and sort of shut down the labs at FIU for students. And so at this point in time, I don't have anything further. Um, However, one of the more encouraging aspects of this is that the PCR of these regions of interest is really the only thing that is a bottleneck in this entire design, because the kit that we get from Illumina is completely agnostic to what the DNA is. It doesn't care. It will tagment the DNA and prepare it for sequencing. The data analysis, we have the ability to identify body fluids and to predict age based off of those regression models. So it's very, very straightforward. The PCR, getting that up and running, is going to be the biggest hurdle in this. However, even there, it's going to be a question of, do I need, you know, perfect amplification of every single one of these markers? Not necessarily. I just need good enough. I need to make sure that I have enough representation of each of these markers.
Thank you very much. I'm going to give the folks a couple seconds to put questions in chat, but one of the things that I wanted to ask you was, I noticed you were talking about how cost effective this is. Have you thought about the total cost to do the complete analysis? I know some of the primers were $10, you were saying, but I was wondering if you thought through that.
So the cost effectiveness that I was talking about earlier was more so for the Pyro sequencing. However, there will be the ability to reduce costs on this as well. So I'm sure most everyone in the lab knows the relative cost of extraction and quantification of DNA. The bisulfite conversion process ends up being $1 or $2 per sample. The bioanalyzer would be about $6 per sample, but that would really only be necessary in the development of this entire process. Once we know that our PCR is working approximately well, you could just use a qubit to get the quantity of DNA that is in our PCR amplicon. And then, so the bulk of the cost would be in the sequencing on the MiSeq, and that's where the ability to multiplex to use barcoding and getting multiple samples into a single run would be hugely advantageous. So the cost to do the library preparation and the sequencing on the MiSeq would be approximately, I believe it was $1,800 in total, but you could do up to 96 samples in that single run. So the overall cost of this per sample, if you batched everything really well, could be, I think it's about $25 to $35. But obviously, at the development stage, it's going to be a little bit higher than that.
It's good, though, from the business perspective and from the lab operations perspective, that you're looking at those costs. $25 versus $2,500 is something that people would like to know before they go through and validating something like this.
Yes, so it would definitely be the use of barcodes to get as many samples in a run would be crucial.
Do you see some of this could be integrated into one smaller system? I know you have a lot of different pieces of equipment for extraction and amplification and those kinds of things. So I was wondering if you saw that there might be a chance to combine some of these techniques into a single microfluidic device or something like that.
So I haven't looked too closely at that. I know that one of the bottlenecks in DNA methylation analysis for now is this bisulfite conversion process that lets us differentiate between the methylated and unmethylated strands of DNA in our sample. And so, unfortunately, it does require pretty good extraction, purification, and quantification before. And also, we're going to need to know about all of the quantity of DNA after our amplification. So, I think we'd be, for the most part, stuck doing this workflow to begin with. I know that the QIAcube, the Easy One Investigator Kit on the QIAcube, I do believe is cheaper than with the Easy One that we have in our lab. Um, and again, you could also pretty just as well use a qubit to quantify beforehand rather than using a Rotorgene Q if you want to save costs in that area. Um, Overall, though, as far as the sequencing is concerned, I'm not sure what other options from other companies would be comparable or less in price.
And since we also have that at FIU, it's always good to use something you have, right?
Yes, yeah. The fact that there's a MySeq just down the hall for me is helpful.
So to the participants, again, if you have any questions for Quentin on his poster, you may want to go back to your poster now,
Yeah,
Quentin. Please put those in chat. We'll try to answer those. Quentin's done one run through. I noticed we have some new participants. We'll wait a couple of minutes to see if we get a few more people, and then he can run through it again. You did a really, really good job, Quentin, by the way, in your first run
thank you.
through. But again, you've got to have some questions. This is very groundbreaking, interesting research, and would like to hear from the participants. Quentin, while we're waiting to see if anybody else has any other questions, have you looked at, you know, how the samples you would look at would be collected from a real world scenario, from a crime scene scenario? Because I noticed you were talking about various different body fluid types, but it looks like they would be potentially collected differently. Uh,
Yes. So the primary way that we collect samples in our lab is on a swab, and so we're extracting from swabs. Um, However, I know that in crime labs across the country, you can be extracting DNA from all sorts of different articles of clothing, even just from uh, a stain on a surface at a crime scene. Um, the amount of DNA needed for all of this is going to be a kind of a hurdle for this research to be applied into the forensic applications. Um, just because it's not so much that you need a certain amount of DNA. Since we're doing a PCR pretty early on in the process, there's always going to be enough DNA, but the amount that we start with uh, is going to dictate how accurate our ratio of methylated and unmethylated strands of DNA in that sample are to the actual individual that it came from. And so we've been doing some studies on uh, looking at the sensitivity of the body fluid identification assay. And the lowest that I currently feel very comfortable suggesting is about 500 picograms. But you're going to get much more reliable results at the 1.5 to 2.5 nanogram input range.
we do have a question for you.
Okay.
Um, the question is, are you seeing any difference in the age estimation from different body fluids from the same donor?
From the same donor? Not necessarily, no. So a lot of these assays have been developed and are pretty accurate from a single individual, as long as you have enough DNA. And that, what I was saying about like the quantity of DNA needed for this to work, this is where it really comes into play. It's the percent methylation that you see is the ratio of the number of cells in that sample that contained a methylated copy of that region or an unmethylated copy of that region. And so the if you have enough DNA The recommendations from QIAGEN for a lot of their Pyro Sequencing work for DNA methylation assays recommends a starting point of 10 nanograms of DNA in the PCR. And that is what's going to make sure that you have reproducible results from the same sample and the same individual. Once you start going lower, you're going to start seeing some variation in the methylation that you get, and that can affect certainly the age prediction.
Very, very good. We've got some new participants, so if you want to run through your poster one more time, just like you did at the top of the hour, then that might generate some more questions. But you're doing great on those questions, Quentin.
All right, thank you. So let's see if I can do it the same way this time. My name is Quentin Gautier. I am a PhD candidate working for Dr. Bruce McCord at Florida International University. My research is primarily focused on the use of DNA methylation patterns for the determination of body fluids that a DNA sample came from, as well as predicting age of an individual based off of their DNA, specifically DNA methylation patterns. So the starting point for this research is the concept that STR kits that have been created by Thermo Promega, they have generated STR profiles with random match probabilities, likelihood ratios that are extraordinarily high, so close to 100%, but we're not going to say that in court, obviously. But what if there isn't any suspect for this crime and there's no hit in our database? Where do we go from there? Some other companies, such as Parabon Nanolabs, they've been looking at creating some phenotypic information, phenotypic profiles for these unknown individuals' DNA that can figure out their skin color, hair color, eye color. Obviously, we can also determine gender based off of melogenin and the STR kits. But still, one thing that we don't have from our DNA is the age of an individual. And this could be hugely important for any investigation where you don't really have a good idea of who the suspect is. You think it might be someone in their 50s, someone in their 20s. We don't really know yet. So what we've been doing in our lab for the past few years is doing Pyro Sequencing of various DNA methylation markers. Now, what this is allowing us to do is to predict or to identify the body fluid from which a DNA sample originated. And separately, it allows us to do some very quick age predictions based off of these CPG results, these DNA methylation markers. However, the Pyro Sequencing platform, although quick and cheap, it doesn't really allow for more than about 4 regions of DNA to be reliably sequenced within the same sample, just for various physical limitations on the instrumentation and the amount of DNA that you're going to get from the whole process. And so what I wanted to do in proposing this research is finding a different method where we're going to be moving on to a much larger, massively parallel sequencing platform like the MiSeq FGX. So what we're going to be doing is looking at a number of locations based off of research that has come out of both our lab and some collaborators around the world, specifically in Europe and Poland, as well as Germany, and then also some collaborators in South Korea. What we're going to be doing is taking our body fluid identification assays and some of our age determination assays and combining it with some other age assays from our collaborators. And the reason that we are combining both the body fluid and the age together is that what research has shown us in the past few years, and if you do a literature search on this area, is that there is a lot of variation on the correlation between methylation and age, depending on what body fluid you're looking at. There are some markers that correlate very well age to methylation to age in blood, but in saliva, it's not as good. And then in semen, it's almost not worth it at all. So that's why we want in the same assay, the very same kit that we're testing this DNA sample from, tell us what body fluid it is, and then we can make the determination of which markers in this assay we pull out that are good for predicting age in that body fluid. To go into more detail about the overall method, the steps that we're going to be taking, the samples can be extracted. In our lab, we use the QIAGEN EZ1 DNA investigator kits, automated, as well as qPCR and our Reuters Q. You could also alternatively use a qubit. And then next is this important step for DNA methylation research, which is the bisulfite conversion step. What this is allowing us to do is to differentially modify our DNA. So what's happening is that the unmethylated cytosines through this process are going to be converted to uracil. That uracil is then going to base pair with adenine during the first cycle of PCR, and then back to thymine after for every subsequent step. Conversely, our methylated DNA, it remains the methylated cytosine remains a cytosine, and so it goes through all of the PCR C to G, G to C, completely as normal. During the development of this kit, we're going to be using the Agilent 2100 bioanalyzer. And what this is going to do, it is a chip-based capillary electrophoresis system that allows for the visualization of DNA, whether it's fragments, PCR amplicons, just whatever DNA you want to put in there, but it doesn't need to be fluorescently labeled at all. You're using a dye. It's kind of like a gel on steroids. What you're going to see are a lot of these peaks here, and this is going to correspond to all of the peaks of DNA that I'm trying to amplify that correspond to either a body fluid CPG site or a age prediction CPG site. Once I'm satisfied with the amplification process and making sure that I have relatively equal representation of each of my target regions, as well as having enough DNA, we can move on to the library preparation step, which is, in this case, using Illumina's Nextera XT DNA library preparation kit. And so this is a very, very quick library preparation. It takes about 90 minutes with only about 15 minutes of hands-on time. And so the way that it works is it's going to take this genomic DNA or amplicons or really whatever you want to throw at it. And it's going to add these transposomes that are going to simultaneously fragment and tag our DNA with these little end cap tags. These are going to be recognition sites for the sequencing primers and indices and the P5 and P7, which are recognition sites on the flow cell for the Illumina sequencers. So once you add these sequencing priming sites, you do another PCR amplification, and then you have your completed library ready to be sequenced. And so once it's ready for sequencing, you add it onto a flow cell, load it into the MySeq FGX, which is going to add it to the flow cell and do that bridge amplification. If you've ever seen one of Illumina's presentations detailing how their massively parallel sequencing works, it is no different. Bridge amplification, you get by interest, and what this is going to allow you to do is get a lot of read depth on each one of DNA methylation sites that you're interested in. Um, Once you've done all of this, you get your data back. And so the interpretation should be relatively straightforward. We're going to have of regions from our PCR amplicons that we're interested in looking at. Once we've aligned, should see is that our CPG sites look a little bit something like this, where at one of these sites, it almost looks like we have a SNP. But what this is that all of these T's that were recorded are unmethylated cytosines, whereas the C's were methylated cytosines. And from looking at the number of C's over the total number of reads that we have at this CPG site, we're going to be able to determine the percent methylation. With percent methylation, we're able to do some data analysis, which starting off with the body fluid identification, this is completely done in-house, this analytical approach here. Looking at these 5 CPG sites, there is our saliva indicating marker, 2 CPG sites for our blood marker, 1 for the vaginal epithelial marker, and 1 for the semen marker. Once we get, for example, this is the profile for semen, we see hypomethylation at 4 of the sites and hypermethylation at this VE8. So what I did was using a process, a statistical analysis called latent profile analysis. What I did was create this profile of what is a saliva sample in the context of getting results on 5 separate markers that are completely independent of each other. So I create a database that tells me what is saliva, what is blood, what is its profile, and then I'm able to plug in these unknowns here. And so these unknowns up here, this is a saliva sample, some blood samples, vaginal epithelial or semen. I test unknown, and what I get back is the posterior probabilities that each one of these samples belongs to one of the profiles that corresponds to a body fluid. And so you can see that we get some very, very high probabilities that these samples are saliva, which is correct, blood, vaginal epithelia, and semen. So, and then you can also see that the probabilities that these samples belong to the other body fluid profiles can be extraordinarily low. Once we know what body fluid we have, we can then take a look at the age prediction models that are specific to that body fluid. And so, for example, from Dr. Lee at our collaborators in South Korea, they have an assay that uses just 3 CPG sites that once you get the percent methylation from these 3 locations, you plug them into this formula and you're going to get a predicted age. And so they reported just a few short years ago about the efficiency of their regression model, having good correlation between the predicted age and the chronological age in both their training set and their test set. So once you get to this point, you're able to produce a report that just based off of these sequencing results, I now believe this to be a semen sample that belongs to an individual that is 52 years old, and there's going to be 52 plus or minus a few years. One way that we envision this being extremely useful for investigators is as different labs are getting used to the idea of genetic genealogy, the idea that you took a crime scene sample, you ran it through a database, a genealogical database, and you have reason to believe that your suspect is a male related to someone in your database, but not that one person. It could be their brother, it could be their father or their uncle, something like that. And so if we now can give you an idea of how old this individual should be, that can help you to either eliminate the younger suspect because it's supposed to be an individual in their mid 50s, or it could even help to include certain suspects that you weren't previously considering. And so I'd mentioned this earlier. I think the first time around that. So this project is supported by the IFRI Student Support Grant. It was proposed back in the fall, in November, I think it was, and it started this spring semester at Florida International University, and I got as far as creating the primers that are going to be needed in all of this before the coronavirus pandemic showed up and shut down a lot of the ability to go into the lab and do research at FIU. However, I'm still fairly confident that this is going to be a pretty straightforward project, given that this technique for the library preparation for sequencing on the Illumina platform is completely agnostic towards what you're putting into it. It doesn't care. It's as long as it is decent quality DNA, it's going to work. So what's actually going to take some time is just making sure that I can amplify all of my targets of interest at a comfortable, reliable level to make sure that I have good representation during the sequencing process. And so that's going to take a little bit of time, but I still think it's going to be pretty straightforward to do once we're able to get back into lab and get work going.
So I do have some questions for you. The first one is, how long does it take to fully complete the MPS analysis, specifically from sample in to answer out, to obtain the body fluid type and the age prediction?
So if we're talking sample in extraction, so the extraction, quantification, and bisulfite conversion process, if you decide to do qPCR, that's about an hour and a half, 2 hours, or if you do qubit, it can be much shorter. But these steps right here, I estimate taking approximately 4 hours total. The evaluation on the bioanalyzer during the creation of the kit, the bioanalyzer only takes about 10 minutes, and afterwards, once you are satisfied with your amplification, you could just use a qubit as a QC check here, so that's even less time. And then the sequencing portion of this, or the library prep and sequencing. So the library preparation kit is approximately 90 minutes total. And then the sequencing on the MySeq would be, I believe it's about 5.5 to 6 hours. So I think the entire protocol, beginning to end, to get your data would be approximately 10 to 12 hours in the lab. And then data interpretation is relatively straightforward. For body fluid ID, you're going to be plugging in the 5 markers for body fluid identification into this model. At least that is how I envision it. And this is calculated very, very quickly. And then the same for the age prediction. So data analysis should actually only take a few minutes per sample.
Okay, thank you. How much sample is needed?
How much sample? So that is one of the great questions for DNA methylation analysis overall. So the amount of input DNA is unfortunately not relevant. So it's not so much the amount of DNA, since we're doing extraction, quant, bisulfite conversion, and then a PCR. The amount of DNA isn't really the issue. The real issue is making sure that the amount of DNA you start with here is representative of the individual. So when we're talking about the percent methylation at any given site or marker, we are talking about the ratio of the number of cells in that sample that contained a methylated copy versus an unmethylated copy of that particular region of DNA. And the less DNA you put into this, the more stochastic effects that you're going to see, the variation in the amount of methylation that you get. For body fluid identification, I've run some sensitivity studies on just this part here, and reliably identifying body fluids down to 500 picograms of starting DNA. And this is in Pyro Sequencing, but it should hold true for this process here on the MySeq. For age prediction, the authors in this paper actually tested this marker and were able to get it to work fairly reliably, remaining pretty close with, I believe it was down to 1.5 nanograms of DNA. But for reference, with the Pyro sequencing that we started with, we were following QIAGEN's recommended protocols, and that one is recommended. They recommend DNA methylation analysis to have 10 nanograms of starting material. So that one is going to be a big ask for forensic laboratories, and I'm sure there's a lot of research going on out there about figuring out different ways to try and decrease the amount of DNA needed to have consistent results.
I have another question for you.
Mhm.
Has there been any research done on methylation with age of sample? Would a sample from 20 years ago have the same methylation, give the same accuracy of age prediction that a sample collected recently would?
Okay, so I do believe there have been studies on aged samples. I can't speak exactly to how old the samples were, but if memory serves, the oldest one in the one I was looking at was about 9 years old, a blood stain on an FTA card. Um, and it gave uh, the age prediction was fairly accurate to the known age of the individual. We've also separately from the age, our lab, Joanna Antunes, who graduated a few years ago from our lab, did a study on aged blood samples for the determination of the body fluid, and found that samples nearly 20 years old had methylation profiles that were consistent with, the known methylation profile of blood. And so if stored properly, it does appear that methylation patterns stay consistent.
Okay, I have another question for you.
Mhm.
With the potential for criminal investigation and eventual courtroom testimony, what type of positive negative controls would you use to show quality assurance, quality control? Basically, how can you show the results are both accurate and reliable?
So, uh, for that, obviously, we're going to be there are some methylation controls that QIAGEN sells alongside their bisulfite conversion kit. And so they will give you DNA that is 0% methylated and DNA that is 100% methylated, and that you can carry on alongside your samples going through this entire process. And so that will be a good control for determining, uh, is there any point along the PCR amplification, library prep, and sequencing that is introducing large amounts of error to the data that you're getting? As far as determining accuracy, that is going to be one of those things that uh, laboratories are going to have to do, obviously, in a validation study, getting a fairly sizable population to contribute samples to create their own database, or separately an entity, maybe NIST, if they choose to start adopting this, similar to the STR tables that they maintain. Um, Basically, this body fluid identification model is based off of the result of 120 samples, approximately 30 per body fluid. And then for these age prediction models, a lot of the time the training sets are based off of 30 samples and then test sets roughly the same amount. And so it's really going to come down to each lab doing a validation study and determining just how accurate this prediction model is for them. And then possibly even teaming up with a biostatistician or uh, really any statistician to figure out if they can refine this model at all to make it a little bit more accurate for them. If, for example, they start using the assay and they're just not getting as reliable age prediction as they would want.
Okay, can you put it back to your poster? We have several new people in the audience. They can review your poster. I have one more question for you, and then we'll see if it generates any more questions from the new folks. This question is, why would you decide to choose the Korean age markers over the other age markers available in the literature?
So the Korean age markers have been reported on in 2015, and then they did a validation study in near the end of 2018, I believe it was, that the mean absolute deviation for their assay is, if I remember correctly, approximately 3.4 years. And so it's one of the models that with just 3 markers, it's one of the more simple assays to amplify and to interpret, and they get extremely accurate results. Um, I've seen some other assays for semen that are not necessarily as accurate or are roughly as accurate, but require many more CPG sites in excess of 10 CPGs. And so I'm looking and hoping to try and make this as straightforward a data analysis problem as possible.
Very good. So any new participants have any additional questions for Quentin as we're nearing about 7 minutes left in our poster session? I want to make sure everybody gets back for 1 o'clock to hear Dr. Lauren Wilson speak on forensic intelligence continuum in the law enforcement context. But we do have time for a few more questions. And Quentin, you're doing a great job in answering those. The people that are asking are even saying you're doing a good job. So
Thank you very much.
I applaud your work. Any final questions as we're wrapping this up so that we can give everybody a chance to take a bio break and get another cup of coffee or a water before our 1 o'clock restart for the. You got a nice thank you there, Quentin, in the chat area. So
thank you very much.
I think what we'll do is we'll go ahead and close this out. I really appreciate your research and doing this poster presentation for us during the virtual symposium. You did a great job, and all your graphics and everything looked good, and your answers were wonderful. So thank you very much.
You.